The disease that we have chosen to study is Greig Cephalopolysyndactyly. Here are a couple of photos showing human individuals who have extra digits, which is a common trait for those affected by the syndrome. Greig Cephalopolysyndactyly is a type of polydactyly, which is a condition in which an individual is born with extra digits on either their hands or feet. The extra digits can be preaxial, which affects the side of the hand or foot with the thumb or big toe, postaxial, which is the opposite side, as well as central, which affects the middle digits. Polydactyly can be further classified into two types. Type A, which involves a full digit, and Type B, which is less than a fully formed digit. Both types can be the result of the same genotype. In general, alleles responsible for polydactyly can show an autosomal dominant mode of inheritance.
Polydactyly is a very common congenital hand abnormality. In a study involving newborns, postaxial type B polydactyly was found in 1 out of every 531 live births. It was also determined that the syndrome is more common in Black individuals than in white individuals. In addition, 76% of cases were bilateral, while 86% of affected individuals showed a positive family history. Polydactyly has also been shown to be more common in males than in females, and while surgical procedures do exist to remove the extra digits, it is often unnecessary and is considered to be more of a cosmetic procedure. Greig Cephalopolysyndactyly is caused by a mutation in GLE3, a gene responsible for a transcription factor of the same name. The GLE3 transcription factor is a zinc finger which binds DNA directly and activates as well as represses transcription of several genes. It belongs to a family of genes involved in the normal shaping of tissues and organs during development and is associated with the sonic hedgehog pathway. It plays a role in cell growth, specialization, as well as a patterning of structures, including the brain and limbs. The function of GLE3 is still not fully understood.
To elucidate the function of GLE3 in humans, we would do a targeted knockout of the homologous gene in mice using CRISPR-Cas9 technology. We would do this by isolating mice zygotes and injecting them with Cas9 mRNA, as well as guide RNA complementary to GLE3. The resulting Cas9 protein and guide RNA together create a double-strand break within GLE3, which will likely result in a mutation upon repair. The developing embryos are injected into a pseudo-pregnant female. Abnormal phenotypes in the pups will give insight to the function of GLE3 during development in a vertebrate model. Next, we want to know what part of the body GLE3 affects the most and at what point during development. We could do this by inserting a visible marker, such as GFP, downstream of the GLE3 promoter and observing what parts of the body have the highest level of expression at several points during development. This would be accomplished in a manner similar to the first experiment, except that now we add a donor vector. The donor vector would be an engineered DNA strand which includes a GLE3 GFP fusion gene surrounded by regions of homology, which would promote homologous recombination and integration of the vector into the mouse genome. This would give us an idea of where GLE3 is highly expressed. In combination, these two experiments would give us a better understanding of what GLE3 is necessary for and how it is expressed during vertebrate development.